So we're still focusing on the question of what exactly those genes things are. And we've established that genes are somehow physically located on chromosomes. A gene is not the same thing as a chromosome, but a gene is, this is connected to chromosomes somehow. There's a lot more genes than there are chromosomes, but it's looking like a gene might be some sort of cluster of chromosomes. And that came from the work of Thomas Hunt Morgan and a number of students of his, including Alfred Sturtevant, Cal Bridges, and some others. And they discovered the phenomenon of sex linkage. And what that demonstrates is that some genes are physically carried on a specific type of chromosomes called the sex chromosomes. And the sex chromosomes come in two types. In mammals and also in fruit flies, we call them X and Y. If you have two X chromosomes, you generally end up female, at least anatomically. If you have an X and a Y, you end up as male. Incidentally, there's no real way you could get someone with two Y chromosomes under the normal course of things, because one chromosome comes from each parent, and a female parent is not going to have a Y chromosome to give because she's female. So you would not normally get YY individuals at all. And if you somehow could, they would probably never be born. There's a lot of genes on the X chromosome that have nothing to do with sex or gender, like that factor 9 hemophilia, for example, that we talked about earlier, that Queen Victoria was a carrier of. And you absolutely have to have those genes in order to survive. You can't really live with no X chromosome. I'm going to kind of skim the details, but Morgan and his students at Columbia University in the famous fly room, the lab that they used that was where they raised many thousands of fruit flies, they went on to show that not only are some genes carried on sex chromosomes, but other genes are carried on autosomes. An autosome is any chromosome that's not a sex chromosome. And Thomas Hunt Morgan, who's shown there on the left, would end up winning the Nobel Prize for Medicine for this work in 1933. Incidentally, his students who'd done a lot of the work did not win the Nobel, although they'd go on to win other awards. But Morgan was kind enough to share his prize money with Calvin Bridges and Alf Sturtevant, shown here as young students over to the right. And I will just point out that Calvin Bridges had really amazing hair back in the day. Sturtevant was able to use a nifty trick called linkage mapping, and we don't have time to go into exactly how that works. Suffice it to say that if you do some crosses just right, you can count how often chromosomes break and recombine during meiosis 1. You might remember that chromosomes in meiosis, but not mitosis, will pair up and then exchange pieces, a process called crossing over. And Sturtevant was able to work out a way to calculate how often that happened, and was able to use that to map the location of genes on chromosomes. We know that genes are located at particular points along the length of a chromosome. And Sturtevant and others who followed him used that frequency of crossing over events in meiosis to construct what are called linkage maps, which show where on each chromosome each gene physically is located. This is a part of a linkage map of the X chromosome of a fruit fly. Every one of these tick marks shows you the location of a particular gene, which is usually named for a prominent allele. So there's yellow body, scute bristles, white eyes, facet eyes, ruby eyes, cross veinless wings, tan body, all of these different traits for eye color and shape and wing shape and body color and bristle shape and so on. And those numbers represent the location of each gene on the chromosome, measured in units that happen to be called centimorgans, of course, after Thomas Hunt Morgan. So chromosomes are these ribbon-looking things. They've got one end and another end, and we can plot where genes are physically located on particular chromosomes. It's actually possible to map the location of these genes. Kind of a neat trick, and I wish I had a little more time to explain how it's done. But we got to keep moving. So what is it about the chromosomes that makes them able to carry genes? The scene shifts. Sorry, I feel like the Golden Girls. Picture this, London, 1928. Some of you will get that. A British bacteriologist by the name of Frederick Griffith has been studying a bacterium called Streptococcus pneumoniae, which causes bacterial pneumonia. That's an infection of the lungs that can be quite serious. And this is something I mentioned way back at the beginning when I was just going over features of, archaea and prokaryotes and all that. And it might have seemed like just some annoying trivia at first, but it turns out that it's important. These are some Streptococcus pneumoniae bacteria seen stained under a high-powered light microscope. And the reason these bacteria look like they've got whitish halos around them is that they're surrounded by a glob of snot, of mucus-like material. That glob is called a capsule. And I know I mentioned earlier that some bacteria, not all, are surrounded by capsules. The reason this matters is that bacteria that have this capsule have a degree of defense against the body's immune system. Bacteria that have that capsule can cause severe disease because they've got a countermeasure against your body's ways of trying to fight the infection. Bacteria that don't have the capsule are less likely to cause disease. They don't have that defense. And you're going to do this in one of the last labs. When you grow bacteria in the lab, usually you grow them on the surface. Could somebody mute, please? Usually you grow them on the surface of the substance that looks kind of like jello.
I was still expecting the national award tuition waiver to hit a few months into my current semester.
Would you please mute whoever that is? I can't really change it from here without stopping the screen share. Okay, thanks. Okay, when you grow bacteria in the lab, you make this stuff that looks like stiff jello and has about that consistency. It's called agar. It's actually derived from a tropical seaweed and sometimes is used as a food additive. But bacteria can grow on the surface of a block of agar without burrowing down into it, which makes them a lot easier to handle. And when you put one bacterium on a blob of agar, sorry, just checking chats and things like that. When you put one bacterium on a blob of agar, that one bacterium will divide into 2, which will divide into 4, which will divide into 8, 16, 32, ... And eventually you get a lump of bacteria because the bacteria can divide. They can make more of themselves, but most bacteria can't move over the surface of the agar, although there are a few that can. And so what you're looking at is what you get when you spread Streptococcus pneumoniae on the surface of a slab of agar gel. S. pneumoniae that doesn't have a capsule, S. pneumoniae can either have the capsule or not. It's variable. And Streptococcus pneumoniae that doesn't have a capsule forms small, rough-looking lumps called colonies, illustrated on the left. And Streptococcus pneumoniae that doesn't have these capsules is called R type. R stands for rough. Streptococcus pneumoniae that does have the capsule.
What does lethargic mean?
Big, glossy colonies that we call S type, S standing for smooth.
Here's what I found.
And again, the S type can cause
What does
severe infections.
lethargic
S type can be
mean?
very dangerous.
Lethargic means affected by lethargy, sluggish, and apathetic.
Please mute.
Oh, sorry.
Quite all right. Right, Streptococcus pneumoniae that forms these big glossy colonies, this S type is dangerous. This is the type that causes severe and sometimes fatal infections. R type Streptococcus pneumoniae, not a big deal. If you get that up your lungs, your body can usually fight it off pretty quickly. And Griffiths found, first of all, that if you inject S-type bacteria in the mouth, the type that does have that capsule, if you inject it into a mouse, not a mouth, if you inject them into a mouse, the mouse will die within 24 to 36 hours. And then if you take a sample of that mouse's bodily fluids and you spread it on a dish of agar, you get live S bacteria. So you inject live S bacteria into the mouse dies, and live S bacteria come out of the mouse. Okay, that's pretty straightforward. If you inject R-type bacteria into the mouse stays healthy, it doesn't die. And if you take some fluids from the surviving mouse, you might get a few R-type bacteria, but you don't get very much because the mouse's immune system has destroyed most of the R-type bacteria that you injected in. Okay, pretty straightforward so far. This is kind of what you would expect. On the left, look at this. You can take S-type bacteria, kill them by exposing them to excessive heat, and then inject them in the mouse, and the mouse will stay healthy. Dead S-type bacteria don't do anything. They don't cause disease. The mouse is fine, and if you take a blood or body fluid sample from the mouse, you get nothing because those injected S-type bacteria didn't grow and didn't reproduce because, well, they were dead. All of this seems like it should be bloody obvious, but here's the kicker. Look over on the right. If you inject a mix of live R and dead S, that does kill the mouse. Think about it. Live R bacteria, which don't have the capsule, they don't kill the mouse because the mouse's immune system can mop them up. Dead S bacteria don't kill the mouse because they're dead. But mixing the two does kill the mouse. And if you take some of the mouse's blood or bodily fluids and spread it on a dish of agar, the stuff we use to grow bacteria, you get live S bacteria. You injected live R and dead S, and what you get is live S. Bacteria do not exactly have chromosomes the way that we'd think of them, but they do have phenotypes, and we've completely altered the phenotype by mixing a live bacterium of one type with a dead bacterium of a different type. That's weird. Griffiths reasoned that there was something that must have passed from the dead bacteria to the living bacteria that transformed them. Something went from the dead S-type bacteria to the living R-type bacteria and turned them into S-type bacteria. And he didn't know what that was, and I seem to recall he didn't really have the time to get deeper into what that was. He just called that substance the transforming principle. Well, that raises the question, what is the transforming principle? Several people worked on this. This was not only the people that I'm talking about, but an American biologist named Oswald Avery, shown here, and colleagues of his started growing Streptococcus pneumoniae in the lab, breaking those cells down, extracting different substances from Streptococcus pneumoniae, purifying them, and testing which one did the transformation. So we take S-type bacteria, grow them, break them down into several different substances, and then test, well, does substance 1 cause transformation? No. Okay, does substance 2? No. Okay, does substance 3? Hey, wait, that does. And so on. And it took 15 years, but they were finally able to isolate and purify that transforming principle. They figured out what the transforming principle was. Turns out that it turned out to be a compound that was rich in nitrogen and phosphorus. If you remember schnapps, you might know that those are the 6 most common elements in life. And the substance was unusually rich in the N and the P in schnapps, nitrogen and phosphorus. It had already been discovered way back in 1869. And it was known to exist in the nuclei of eukaryotic cells. And it turned out to be weakly acid, not nearly acidic enough that it could burn you or anything like that. In this test tube that you're seeing right here, it's the slightly purplish stuff that is floating right in the middle of the test tube. It's this. That's a lump of this substance that, since it's weakly acid and is found in the nucleus of eukaryotic cells, as well as being found in prokaryotes, it's called nucleic acid, sensibly enough. And it turns out that one of the substances that chromosomes are made of happens to be nucleic acid. The other substance that chromosomes are made up of is called protein, and we'll talk later about proteins. Suffice it to show that Avery was able to show that protein was not the transforming principle. Protein by itself did not cause bacteria to change their phenotype. What does cause that change is nucleic acid, which is found in prokaryotes and eukaryotes as well. What the heck is nucleic acid? You probably know some of this stuff already if you had chemistry in high school, but let me backtrack just a little bit. Matter, that's everything that's got mass and takes up space, solids and liquids and gases and all of that, is made up of extremely tiny particles known as atoms. As we say, never trust atoms because they make up everything. That's a joke. I'm imagining you all laughing uproariously right now. In the diagrams we're going to in the diagrams that we are going to look at, atoms are generally depicted as little colorful ping pong balls that stick together. That's not really what atoms are. Atoms, of course, turned out to be made of protons, neutrons, and electrons. If you want to know more about what atoms really are, we've got a perfectly good chemistry department that can set you straight on that. For what we need to do here, we can pretend that atoms are little balls that stick together. It's not the most accurate picture, but it will do at the level of understanding that we've got time for. There's 88 naturally occurring types of atom. In other words, there's 88 natural chemical elements. There are also over 30, something like 30, what are we up to? 118. And then there's a couple more. There's a bit over 30 elements that have only been synthesized in laboratories. Some of those actually have commercial uses. Others have never existed in more than a couple of atoms at a time. We don't really need to talk about them for our purposes here. But there's 88 stable types of atom that can be found in nature. There's 88 natural chemical elements, including, of course, hydrogen, nitrogen, oxygen, carbon, phosphorus, and sulfur, and then everything from gold to mercury to iron to praseodymium, all of those. And we don't really need to cover why they do this or what happens, but atoms can stick together. Some atoms are much better at doing this than others. Atoms of helium are almost impossible to stick to anything else. Atoms of chnops stick to each other quite readily. And when atoms stick together more or less permanently, we call that forming a chemical bond. And when you have two or more atoms sticking together, we call that cluster a molecule. And when we have two or more types of atom, two or more elements that are bonded together, we call that a chemical compound. That was very basic, and I'm assuming most of you have at least been exposed to that in the past. But I just want to make sure we're all starting from the same point. This is a diagram of one molecule of a chemical compound called styrene. You have every one of these letters C is an atom of carbon. And you can see that there are 6 carbon atoms that form a ring. Each of these black lines represents a bond between 2 atoms. And you'll notice that some of these bonds are single and some of those bonds are double. You can get this, especially with carbon, it happens a lot, that you can have a double bond or even sometimes a triple bond between 2 atoms. And again, for our purposes here, we don't really need to worry about why. The carbons are chained together in a ring, and there's a couple of carbons sticking off the ring. And then sticking off the carbons, we have various atoms of the chemical element hydrogen. Okay, that compound is called styrene. If you take several styrene molecules like this, it's actually possible to do something really nifty. You can make them start linking together in a long chain where each styrene molecule connects to the styrene molecule in front of it. And then a new styrene molecule can connect to that one, and so on. The chains can get longer and longer and longer, and you end up with something that looks like this. If you take a bunch of styrenes and string them together like this, you get a long chain-like molecule. This is just a small portion of one called polystyrene. And polystyrene is used for everything from disposable plastic cups to disposable plastic knives and forks and spoons, to the jewel cases that CDs were kept in. If any of you remember back before the internet days when we bought music on CD, those cases are made of polystyrene. And then polystyrene with a lot of air bubbles added to it is called styrofoam, and so on. Polystyrene is a great example of a polymer. And in fact, plastics are polymers. If instead of stringing a bunch of polystyrene molecules together, we strung a bunch of vinyl chloride molecules together, we would have a different type of plastic called polyvinyl chloride, aka PVC. I assume you've seen PVC pipes at some time in the past. Maybe your house has some plumbing done with PVC pipe. And PVC would be another example of a molecule that's built out of many little subunits strung together in a long chain. And any big molecule that's built that way as long chains of subunits is called a polymer. The subunits themselves are monomers. So in the case of polystyrene, the chain, the polystyrene molecule is the polymer. Each one of these styrene molecules that are stuck together to make it up are called monomers. Plastics are great examples of polymers. So are artificial fibers like nylon. And many biological molecules are also polymers. And you can think of a polymer as kind of like a train. You have a number of subunits, like the cars on a train, that can be strung together in a series of hundreds or maybe more than that. Like a train, some polymers might consist of more than one type of subunit. In the polystyrene that we were looking at, all of the subunits look the same. They're all styrenes stuck together one after the other. But some polymers, you might have more than one type of subunit, just like a train might contain more than one type of car. You might have a mixture of box cars, flat cars, those piggyback freighters that they, with the big boxes on the back that you can stick right on the back of an 18-wheeler. There might be tank cars. There might be, well, these days they don't mix passengers and freight, but in the old days there might be passenger cars or cabooses or things like that. And with just a few types of different subunit, boxcar, flat car, tank car, whatever, you can make a huge number of possible combinations. Think about that the next time you get stuck when a train is passing through downtown Conway. As that train rolls past, you might ponder that there's a small number of train car types, but they can be strung together to carry just about any load you can think of. And polymers turn out to be incredibly important in the chemistry of life. What you're looking at here is a molecule of cellulose. And you might remember I said a long time ago that the cell walls of plants are made of this stuff called cellulose. By the way, humans can't digest cellulose, so it passes right through our digestive system untouched. And when we eat it, we refer to it as dietary fiber. So if you've ever eaten like a high fiber cereal, like, Kellogg's All Brand or something, or, a nice high fiber bran muffin or something like that, you've been eating lots of cellulose. And what cellulose looks like on the molecular level is each one of these things is a ring of carbons. They're not drawn here for clarity, but at every one of these angles, there is a carbon atom, and I'm drawing them in right now. And there's also some hydrogens that for clarity aren't shown. I'm not going to draw all those in, but there's some hydrogens stuck here. And this is a ring of carbons, hydrogens, and oh, look, there's an oxygen just for flavor. Each one of those rings is a simple sugar. Take a bunch of simple sugars and string them together in a chain like this, and you get starch. String them together in a longer chain in a slightly different way, and you'll get cellulose. String a slightly different sugar, which I think is N-acylglucosamine, don't quote me on that, but string a slightly different sugar together in a long chain and you get chitin, which I told you a long time ago was what the cell walls of fungi were made of. So starch, cellulose, and chitin are all polymers of simple sugar molecules strung together in a chain. We will talk a lot more coming up about proteins, but I know you did those chemical tests in lab for the presence of protein in foods. And proteins are polymers of simpler molecules called amino acids, and I'll talk about those later. Those of you involved in nutrition or conditioning or things like that probably know about essential amino acids and so on, and we'll get to those. And in 1919, a guy named Phoebus Levain described the chemical structure of the monomer that nucleic acid was made of. Okay, somebody just typed their name. Nucleic acid turned out to be a polymer, and the monomer that makes up nucleic acids turns out to be something called a nucleotide. And you're looking at a diagram of a typical nucleotide. And just as a train car has parts like the wheels and the base and the hitch and all that, nucleotides are made up of parts. So a nucleic acid molecule may be made of nucleotides. Each nucleotide is made up of parts. Over here, shaded in kind of beige, you have an atom of the element phosphorus stuck to 4 oxygens plus a couple hydrogens for flavor. And we call that grouping of atoms the phosphate group. A phosphorus stuck to 4 oxygens is called a phosphate. The phosphate is stuck to, highlighted in green, a simple ring of carbons with a couple of oxygens. And again, carbon is so common, we don't draw it in formulas, but every one of these angles right here is the site of a carbon atom, and there's various hydrogens stuck on here as well. And that's a simple sugar. And then stuck to the sugar is either a single ring, or in this case, a double ring of carbon and nitrogen atoms, and we call that the base. So every nucleotide consists of a phosphate group, a simple sugar, and a base. Okay, if we had an extra oxygen stuck right here to this carbon with a hydrogen glommed on it, we would have a sugar called ribose if we had that extra oxygen. Turns out we do not, in fact, have that extra oxygen. It's not there. And so the sugar right here is not called ribose, but because it's missing its oxygen, we call it deoxyribose. Ribose with an oxygen taken away. So there must be hundreds of different types of simple sugar. Some you might know, like fructose in that high fructose corn sweetener in our diet, or glucose, the sugar that diabetics have to monitor. This one is ribose, except it's missing an oxygen, so we call it deoxyribose, ribose with an oxygen removed. And a polymer of nucleotides that contain ribose is called deoxyribonucleic acid. Or if you don't want to say deoxyribonucleic acid, you can just call it DNA. Now we're getting somewhere. DNA is that transforming principle and it's a polymer of nucleotides. And in fact, unlike cellulose and starch and plastics, where there's only one type of monomer, DNA consists of 4 different monomers strung together. They've all got a phosphate group and a sugar group, but each type has a different base. So the base right here at the upper left, highlighted in pink, is a double ring of carbon and nitrogen atoms and a few hydrogens that aren't shown. And that double ring is called adenine, just abbreviated A. For the record, you do not have to memorize the formulas for any of these. I would expect to show you a picture like this, and you could identify which one's the phosphate, which is easy because it's got a P in it, which one's the sugar, and which one's the base. And the base always has lots of nitrogen atoms in it, so it's pretty easy to identify. But I would not expect you to memorize the specific formulas of these 4 bases. But this one up here at the top left is adenine. This one down here has a slightly different base, although it's still a double ring of carbons and nitrogens, and in this case, a little oxygen's got in there. That base is called guanine. At the upper right, we have a single ring base called thymine. And down here, the 4th possible base is also a single ring of carbons, nitrogens, and an oxygen stuck on, called cytosine. So we've got 4 different bases: adenine, guanine, thymine, and cytosine. Those 4 bases are parts of different nucleotides. All nucleotides have the same phosphate and sugar groups. If we're going back to trains, it's a little bit like all train cars have the same wheels and the same wheelbase and the same latches to link them together, right? It has to be that way, otherwise trains couldn't run on tracks, right? If different cars had different spacings of the wheels or different sizes of the wheels, the train's not going to be able to stay on the track. So all trains in a given country have the same basic wheelbase and the same type of latches, but what's on top of that might be different, right? There might be an oil tank, or there might be a boxcar compartment or a refrigerated compartment or whatever. So just as all train cars are going to have the same wheelbase, but different cargoes on top, all nucleotides have the same phosphate and the same sugar, but different nucleotides on top, as it were. And there's 4 different nucleotides that are used in DNA: adenine and guanine, and thymine and cytosine. Okay, last thing before we finish. A biochemist in Austria by the name of Erwin Chargaff discovered a few facts about DNA. First of all, if you break down DNA into its nucleotides, it turns out the percentage composition of nucleotides is different in different species. Your DNA turns out to be roughly 29% adenine. Corn DNA, it's about 27%. Octopus DNA, it's about 33%, and so on. You know, you might expect that if DNA is this transforming principle, if DNA is the stuff responsible for heredity, DNA makes you look like what you look like, then you might expect that there would be, in fact, chemical differences between your DNA and octopus DNA. Your DNA is not identical to octopus DNA because you're not an octopus. Duh. I hope nobody, if anybody is an octopus, I apologize. I'm not trying to be insensitive here. So that might not be surprising, but Chargaff noticed something. He noticed that the percentage of adenine was always equal to the percentage of thymine, and the percentage of guanine always equaled the percentage of cytosine. This is a very common type of test question that I give. If you know the percentage of one of the nucleotides, you can work out the others. So let's take human DNA. Human DNA is 29% adenine. The percentage of adenine is always equal to the percentage of thymine. So we know it's also 29% thymine. What about the others? Well, there's only these four. So the percentages of adenine, thymine, guanine, and cytosine must add up to 100. So the percent of guanine plus cytosine must equal 100 minus 2 times 29%. Okay, 29 plus 29, or 2 times 29 is 58. So 58% of the DNA is A and T. 100 minus 58 is 42. So 42% of your DNA is G plus C. And since the percentage of guanine always equals the percentage of cytosine, then you must have 21% guanine, because that's half of 42, and 21% cytosine. Sorry about the rotten handwriting. Hopefully everybody got that. So if you know the percentage of one of these nucleotides, you can calculate the percentage of the other three. And that's a very, I like asking exam questions about doing this. So you might want to remember that. And these arithmetic rules that we find are still called Chargaff's rules to this day. Why do they work? Why is this true of DNA? We'll have to get the answer on Friday. So with that, I will leave you to contemplate Chargaff's rules, and I will stop the recording.